So in this video what we're going to have a look at is independent segregation in a bit more detail. Remember, this is one of the mechanisms that is generating genetic variation in the daughter cells and the gametes that are produced by meiosis cell division. I'm going to use the same colours representing chromosomes as was used in the overview of meiosis. So that's orange, green, orange-brown rather, and a light green and a dark green. So we need to just remind ourselves that in the cell we've got, in this example, two homologous pairs of chromosomes, and we can tell that they are different chromosomes because they're of different lengths. Remember, the different colours within each homologous pair, orange, brown, light green, dark green, is just there to represent the fact that one of those chromosomes within a homologous pair is maternal and one is paternal. So towards the end of interphase, each of those chromosomes undergoes a semi-conservative replication. So we end up with that, and with that taking place in the other homologous pair of chromosomes. The point at which independent segregation occurs then is within metaphase 1, and this is going to lead to the different variation within the gametes. Remember, I want you to be able to use the word bivalent when we are now discussing the movement of these homologous pairs of chromosomes. So remember, we can't see this taking place in late interphase, but it is occurring. We move into prophase 1. The homologous pairs of chromosomes pair up, and then they form bivalence. What I'm representing then here then is we're going to look at metaphase 1. So if we start off by looking at the left-hand side cell just here. So I'm going to have the equator running vertically. Remember, this is completely random. So if we look at our smaller bivalent, purely by chance, it could be that the bivalent arranges itself like so: orange left, brown right. The other bivalent, the larger homologous pair of chromosomes, they will move and arrange themselves at the equator independently of the other bivalent, and they will segregate, they will arrange themselves independently of this of the other one. So it might be, and this is all just purely by chance, that the other bivalent arranges itself like so. So this is within metaphase 1 of mitosis. The point is this, is that in the same organism, let's say that we're looking at gametes, ova being formed in ovaries within a female, in a cell right next to this one, so this can't be the same cell, but it's in the same organism, due to the fact that these bivalents arrange themselves independently of one another, we could have the following taking place. We may have the smaller bivalent still arranging itself like so, but purely by chance the other bivalent arranges itself. So now we have the dark green chromosome on the left hand side and the lighter green on the right. So what's the significance of that? Well, We know that we'll run through then anaphase and telophase, and so the arrows are just representing the direction in which the chromosomes are going to be separated to opposite poles. What this independent segregation then leads to is very different possible combinations of chromosomes ultimately within the gametes. So let's just look at our first scenario here, if you like, on the left hand side. Let's look at scenario A. Remember, once we move into the second meiotic division, the spindle fibres arrange themselves at right angles to where they were previously, and so in this case now we're going to have the equator running from left to right. So once our first cell division has occurred, and we've now moved into, this is basically metaphase 2, is that in one of these cells from the first cell division, we're going to have this arrangement, and in the other cell we will have this arrangement. Once we run through into the second cell division, which is going to occur at right angles to how it did previously, again, let's just focus on this particular arrangement of bivalence, as we can see here. That will then lead to 2 new cells formed from each of these, and so we have a total of 1, 2, 3, 4. So we'd end up with the following possible combination of chromosomes. So that's what we could get. We could get small orange, large light green, or we could have small brown, large dark green, and that's replicated here. But these are the two different types of chromosome combinations that we could get. If we look at our other scenario, let's call this B then. So now we're looking at the fact that the larger bivalent has arranged itself differently to A. We're going to get a very different combination of chromosomes in the gametes formed. So we have small orange, and now we've got larger dark green, and we have small brown, and we have larger light green. Remember, then we're going to have our second cell division, and the 4 gametes are going to be formed. You can see that we now have a different combination of chromosomes. Again, these are the 2 different combinations of chromosomes, and that's it really. So independent segregation then has this ability to generate different combinations of the maternal and the paternal chromosomes that are available to them. I've included a link to another animation which runs through this with you.